Assalamu alaikum dear students. I am Muhammad Rauf. Welcome to the course Principle in Animal Life 2. This is lecture number 29. The topic is Recombinant DNA Technology. Discussing agenda for today's topic is Introduction, Discovery of Recombinant DNA, Steps in Recombinant DNA Technology, Gene cloning purposes, vectors, sources of gene of interest or donor DNA, restriction enzymes cut DNA, ligase joins DNA, lecture outcome. After watching and listening this lecture, students will know about various steps involved in recombinant DNA technology. Introduction. Recombinant DNA is formed when scientists combine pieces of DNA from two different sources, often different species, in vitro to form a single DNA molecule. Means you have two organisms belonging to two different species and you collect DNA molecules from both of them and then you join them with the help of enzymes. Then the product, the hybrid DNA product which is formed is called recombinant DNA. In molecular biology, cloning means the amplification of any DNA fragment through recombinant DNA technology. Cloning means making multiple copies of a particular DNA fragment. If the fragment is well-defined, complete gene, the process is called gene cloning. Gene cloning or DNA cloning is a process in which several copies of a gene are synthesized, just like we have a process called PCR where hundreds of thousands of copies of a DNA strand can be synthesized in vitro. Here hundreds of thousands of copies of a gene is synthesized in vivo inside a living system. Although it may seem like a modern field, biotechnology, the manipulation of organisms or their components to make useful products, actually dates back to the dawn of civilization. After 1970s, the recombinant DNA technology brought a revolution into that area. But this area dates back to ancient times when the people used to make beer and bread from yeast and selective breeding of pets to produce organisms having better phenotypes. As I already said that this area is one which began from the dawn of civilization, but a revolution was brought about in this field after the development of recombinant DNA in 1970s, and then the terminology shifted from biotechnology to DNA technology. Now, using the these methods, scientists can, for instance, extract genes from one organism and transfer them to another, effectively moving genes between species as different as E. coli, bacteria, papaya, and fish. Now there is so much development in this area that we can combine DNA molecules from two species which are completely different from each other. For example, a gene taken from a fish can be incorporated into a prokaryote E. coli. Discovery of recombinant DNA. It was back in 1972 when Paul Berg developed the first recombinant DNA, and he inserted viral DNA into a bacterial DNA. Then, after this discovery, the attention of several other scientists also diverted towards that area, and in 1973, Herbert Boyer and Stanley Cohen introduced genes from the toad Xenopus laevis into bacteria and showed that the genes could be passed from generation to generation and were expressed in bacteria. Initially, Paul Berger inserted a DNA fragment from virus into bacteria, but then in 1973, Herbert Boyer and Stanley Cohen moved one step ahead and they introduced a eukaryotic gene into a prokaryote, and furthermore they were successful and successfully observed the expression of that eukaryotic gene in a prokaryotic system. What are the different steps in recombinant DNA technology? A bacterial plasmid that will serve as the vector or gene carrier. Bacteria has small circular extrachromosomal DNA molecules that are called plasmids, and plasmids are DNA molecules that doesn't wait for the bacterial DNA to replicate. They can replicate on their own. Their replication is independent and they can produce hundreds of thousands of copies. They have their own origin of replication. They have an antibiotic resistant gene and they have a selectable marker. So the plasmids are taken out from bacterial cells and then the gene of interest which is to be incorporated into the plasmid is either taken from an organism or an already developed cDNA library or genomic library. Then these genes from the source needs to be cut from the source and a slot should be created in the plasmid to incorporate that gene of interest. Now both those processes needs some enzymes, some molecular scissors, and these molecular scissors are called restriction enzymes that cuts DNA at specific locations. These are called restriction enzymes because each of these enzymes has their own special sequences where they initiate a cut, a double-stranded cutter. Its restriction enzymes were initially discovered in bacteria and they are part of a defense system in bacteria. Whenever bacteria is attacked by the viruses, these restriction enzymes are activated and they defy the attack. So the restriction enzymes cut DNA from both sources. The plasmid and target gene are mixed. The single strand ends of the plasmid base pair with complementary ends of the target DNA. Means once the restriction enzymes produces a cut in the plasmid, then this area is used for the incorporation of the gene of interest, and then ligase enzyme is used to establish a bond, a covalent bond between the plasmid and the newly inserted gene of interest, and the resulting molecule, the resulting plasmid is called recombinant DNA molecule. Then the recombinant plasmid is introduced into the bacterial cell through a process called transformation, and then the bacterial cells are selected for whether they have the recombinant plasmid or not, and for this purpose the selectable marker which is used is usually an antibiotic resistant gene which is present in the plasmid. All the bacterial cells are exposed to an antibiotic, and only those cells survive which have the recombinant plasmid and the uh, antibiotic resistant gene. Here you can see the process. This is a vector, a plasmid, and this is the gene of interest. The vector is cut with the help of restriction enzymes, and when a slot is created, it is usually filled by the gene of interest. Ligase enzyme is used to seal the ends, and then this recombinant molecule is introduced into a bacterium, and if this transformation is successful, then the plasmid has its own origin of replication. So the plasmid replicates independent of the bacterial DNA, and in a short span of time thousands of copies of the gene of interest are synthesized. Here is the same illustration in a more detailed form. This is a bacterial cell, and here its plasmid. The plasmid is removed. It is cut with the help of restriction enzymes. The gene of interest from other sources introduced, and then this recombinant plasmid is introduced into the bacteria, and then the objective of the experiment is achieved. Either the objective is to extract the protein for which the gene of interest codes, or to make multiple copies of the gene of interest. Here they have inserted a gene which is responsible for producing some kind of protein like growth hormone, or protein that dissolve blood clots in heart attack therapy, or a big protein that degrades the toxic wastes, or a gene for pest resistance. So it depends upon the objectives of your experiment. If the objective is to produce multiple copies of the gene, then the vector is called cloning vector, and if the objective is to produce the protein for which the gene of interest codes, then such type of cloning is called expression, and such type of vector is called expression vector. Cloning purposes. Gene cloning can be used for two basic purposes: to make multiple copies of the gene of interest, to harvest and use the protein product of the cloned gene, insulin vector. Now, vectors are the vehicles that carry the gene of interest into an organism. Vectors can be physical, they can be chemical, they can be biological. Here we have already discussed a biological vector, that is plasmid. The choice of the vector depends the choice of the vector and the choice of the organism in which the recombinant DNA is to be introduced depends upon your objectives, what type of gene of interest you are going to incorporate into the vector, whether that gene of interest is a complicated eukaryotic gene which cannot be expressed in bacteria. So the choice of the expression system and the vector depends upon your gene of interest. Plasmids are ideal vectors. Why they are ideal? Because the transformation process is easy. They are smaller in size. They replicate independently. They have antibiotic resistant gene which is used as a selectable marker. So they have all the features, but the problem with the plasmid is that they can only accommodate a very short a gene of interest of very small size. So the large eukaryotic genes cannot be cloned using plasmids. They are cloned using other types of vectors like bacterial artificial chromosomes or bacteriophages or other vectors. Now the gene of interest is incorporated into the cell through electroporation, through gene gun, through liposomal delivery. As I already said that the mechanism of introduction of the gene of interest into the expression system varies, and it can be chemical, physical or biological. Now from where the gene of interest is going to come, the scientists usually develop two type of libraries. cDNA library, where the messenger RNA is taken from an organism. That messenger RNA is usually in a mature form and it does not have the non-coding regions or introns, and then through this messenger RNA the nucleotide sequence is determined, and the cDNA library is synthesized. So the cDNA library is usually a smaller because it doesn't has the non-coding areas, and a gene taken from cDNA library can be incorporated into a system which lacks the ability of post-transcriptional modification or splicing. On the other hand, the genomic library is a large library, and the gene in that library also has the non-coding regions. Here we have restriction enzymes, as already said, that they were discovered in bacteria and they are part of a defense system in bacteria. And restriction enzymes are called so because they have a specific site at which they make a cut. You can see here this is a sequence of nucleotides GAA, TTC, and here it is making a cut in that particular area. So it is producing sticky ends. These are sticky ends, and this one, the straight cut, it produces blunt ends. Sticky ends are more favored in molecular biology techniques. And Werner Aber discovered restriction endonuclease in bacteria. And E. coli became the first commercially available molecular scissor Why it is called E. coli, why it is named so, because it was obtained from Escherichia coli. R stands for restriction enzyme and I means 1, type 1. Here you can see an illustration. So this is an enzyme and it is cutting that particular sequence and it is producing sticky ends. And then this is the gene of interest. The gene of interest is also cut with the same enzyme, and then this gene of interest is incorporated into that slot, into that sticky ends, and recombinant DNA is synthesized. Here is another illustration. That's the end of the lecture. Thank you very much.